Okay, so ladies and gentlemen, you are all welcome, and today we are going to look at the uh, enzymes, and that is our today's lecture. So let me share my slides. Let me share my slides. So you are all welcome, and today we are going to look at the concept of enzymes, and this is almost we are almost done with the course. And I think after enzymes, the next thing that we are going to discuss on is genetic material, which is, I think, basic of central dogma. So let's actually try to concentrate on the concept of enzymology, and you are all welcome to the YouTube channel. And of course, if this is the first time you are coming, sorry, if you didn't actually join the YouTube channel, let's try to join the channel. As after this of our classes, I'm going to upload videos to the members of the team, especially maybe you will get some first question paper and solve them. So I hope you can all see my slides. Can you all see these slides? Can you all see the slides? Okay. So what are enzymes? So enzymes actually are biological molecules that speed the rate of biological reactions. So we have a catalyst. Remember from our basic knowledge of chemistry, we have a catalyst. And when we said a catalyst, it means that are the chemical substances that speed the rate of a chemical reaction. And we have different types of catalyst. We have organic catalyst and we have inorganic catalyst. So organic catalysts are enzymes. And what are enzymes? Enzymes are biological molecules that speed the rate of biological reactions. Generally, in living organism, in a human being, like in human being, in every seconds, there are over 1,000 reactions that are taking place in our body. There are over 1,000 reactions that are taking place in our body. And each of these reactions are catalyzed by enzymes. Each of these reactions are catalyzed by enzymes. So what are enzymes? So enzymes are biological molecules that speed the rate of the reaction. And when we say they speed the rate of the reaction, generally there are some reactions in our body without enzyme they can take hours, they can take years. You assume yourself now you eat carbohydrate foods. You assume yourself that you eat carbohydrate foods and that carbohydrate is not digested or is digested maybe after 2 days or 3 days. It means that energy will not be generated. And if there is no energy in your body, there will be improper functions in the body. So you need enzymes so that to speed the rate of the reactions so that all the physiological process will be maintained in the body. So that is one of the most important aspect of enzymes. And in addition, enzymes are proteins. Enzymes are proteins, but very few of the enzymes are nucleic acid in nature. So there are proteins, and remember we talk about proteins, and proteins have different level of structure. We have different levels of enzyme structure or protein structure. We have primary structure, we have secondary structure, and we have tertiary structure and quaternary structure. So enzymes, they usually exist at the level of tertiary and quaternary structures. So enzymes exist both as tertiary and quaternary structures. So they have both secondary and quaternary structure. It means that some enzymes exist at tertiary structure, while some enzymes exist at quaternary structure. And one very interesting thing that I want us to understand, whenever the quaternary structure or the tertiary structure of proteins is lost, it means that the function of that particular protein will also be lost. And I think I gave you this example. We know that snake venom is very deadly, it's very dangerous. So whenever there is a bite from a snake, it depends on the content, it depends on the protein content or enzymes content of the snake. Like some snakes have hemorrhagic effects. They have hemorrhagic effect. When we say snake have hemorrhagic effect, it means that they affect blood, and some snake have hematotoxic effect. That is, it affects the actually the red blood cells. That is hematotoxic. That is affects the blood cells in the body. And some venom of the snake have neurotoxic effect, and it's because they affect the nervous system. It affects the nervous system. So, but now the question is, what make a snake venom to have effect on the nerve cells? What make snake to have effects on the erythrocytes? What make blood to have effect on the white blood cell is based on the protein content. Like for example, some snake venom are highly rich. They are highly rich in phospholipase A2. And phospholipase A2 always affects the membrane of a cell, that is the cell membrane. So it means that it can have hematotoxic effect. So based on that is because of the protein content. So if the tertiary and the quaternary structure of the protein is lost, then the activity of that enzyme should also be lost. So you should understand that. So enzymes can exist at either at a level of tertiary structure and quaternary structure, and they act as a catalyst, meaning that they accelerate the reactions in the body. They accelerate the rate of the reaction in the body. And so that enzymes will still remain itself, it will recycle itself to go back and convert another substrate to product. So enzymes are not consumed during the course of the reaction. They are not consumed during the course of the reaction. So enzymes are not permanently, and that is what you call enzymes. So this is actually a typical example of enzymes. This is a it's a typical structure of an enzyme. This is an enzymes, and this site called active site, and we are coming to that. So enzymes usually they are very specific for what they catalyze, and they are reusable, and usually their names end with ASE. So what I'm saying here is enzymes generally they are very specific on what they catalyze. Okay, it's like someone is saying that you cannot see the slide again. Is that true? But here to me, I think it's very clear. Are you seeing the slides? Okay, so let me share the slide again. So I think you can now see the slide. So what we are seeing, enzymes are specific for what they catalyze. So if enzymes is catalyzing only glucose, so it cannot catalyze protein. So that is why they are specific. Even in the, like for example, carbohydrate, in the carbohydrates, we have what we call monosaccharide and disaccharide. So under monosaccharide, example of monosaccharide, we have fructose and we have glucose. So if glucose is catalyzed by enzymes A, so it's that only enzymes is that only glucose that will catalyze by that enzymes. And if the enzymes is catalyzing protein, let's say glycine, so it's only catalyzing glycine. So that is actually what you call an enzyme. So enzymes, they are highly specific on what they catalyze and they are reusable. It means that if enzymes is used to convert A to B, if enzymes is used to convert A to B, then after it convert that A to B, you can still use it again. So that's the meaning of reusable. You can use it and reuse it again. So that is what you call enzymes specificity. It is highly specific and it can be reusable. You can reuse the enzymes, use it again, use it and reuse it. And that is one of the most important things with enzymes. And then in addition, enzymes always act on a substrate, and before that also enzymes usually end with ASE. Enzymes are usually end with ASE. Like for example, sucrase is an enzyme, lactase is an enzyme, maltase is an enzyme. So enzymes usually end up with ASE. They usually end up with ASE. Although there are some enzymes that their names are not ended with ASE, but that was actually happened before the standard way of naming enzymes. Like for example, pepsin, trypsin, they are all enzymes. They are not ended with ASE. So sucrase, lactase, and maltase are the example of enzymes that are now end with ASE. So it's not all enzymes that end with ASE, but in a current system of classification of an enzymes, they end with ASE. They end with ASE. And these enzymes, they usually have an active site where a substrate binds. So for example, here this is the active site of an enzymes, and this is the substrates that bind to the active sites. So before actually enzymes converts a substrates to product, the enzymes usually work by weakening the bonds. It weak the bond of the substrates, which result in lowering the activation energy. And remember from our basic knowledge of chemistry, when we said activation energy is the energy barrier that must be overcome before reaction takes place. So what actually happens is before a substrate is converted into product, it must overcome that activation energy. It's like a hill that the substrate must jump before product is generated. So in the case of enzymes, what they do actually, they weaken the bond through lowering activation energy. Then you have the reaction. So the reaction will take place. So that is what actually, or that is how enzymes work. It work by lowering the activation energy. So in addition, this is what we call an energy profile. We have reaction progress or progress of reaction. So we have free energy against progress of the reaction. And we have our reactant and product. So without an enzymes, this is what we have without an enzymes, and this is our activation energy. So without an enzymes. But with an enzymes, you see that the activation energy is lower. So meaning that when activation energy is lower, it means that the substrate can easily be converted to generate product. I hope you are all hearing me and also you are seeing the slides. Are you all seeing the slides and you are also hearing me? So the enzymes are usually lowering the activation energy. So it's like a hill that the reactant must overcome before the reaction reach completion. So enzymes usually lower the activation energy. And once the activation energy is lower, then the reactant can easily be converted to generate a product. So especially during what you call reaction mechanisms, before reaction takes place, the reactant particles must collide with one another head to head to generate a sufficient energy that will make them to jump the activation energy. And if the activation energy is jumped, then the product will be generated. But if the reactant particles do not generate enough energy, it means that they cannot be converted into product. And that is what usually happens. So you have an enzymes. The enzymes usually ease the substrate to be converted to product by lowering the activation energy. And that is what it does. Substrates and the products, enzymes and the reaction. So without an enzymes without a substrate, this is an enzyme here. This is a normal base. This is a normal what, or this is what we usually do in the lab. So you see, enzymes without a substrate. So it means that no reaction. If you have an enzymes and there is no substrate, it means that no reaction will take place. There will be no reaction that will take place. And then here you have enzymes plus substrates. So if you now observe here, you will observe that you will see that there is a bubbles. A bubbles is formed. So you see enzymes plus substrates, water molecules and oxygen are generated. And at the end, you have a bubbles. You have a bubbles. So this is to show you that there is an enzymes and a substrate. So there must be an enzymes and a substrate before reaction takes place. Ladies and gentlemen, the aspect of enzymology is very, very important in the area of biochemistry and in the area of clinical sciences, because enzymes, it is very, very important in terms of drug discovery, disease diagnosis, in terms of, apart from disease diagnosis, in terms of disease treatment, in terms of disease marker, you can only use enzymes. So that is why it is very, very important to understand the substrates and the enzymes. The substrate is for which enzymes and the enzymes is for which substrate. So it is very, very important. Okay, Akinola Kehinde, you are all welcome and for joining the YouTube membership. So that is why I think this is where you will really appreciate the concept of biochemistry, because we are going to look at the application. In fact, your assignment, your group assignment will center on this aspect of enzymology. So that is why it is very, very important you should understand the concept of enzymology, so that at least when you are doing your assignment, you will do the right thing. So what we are saying actually, you must have an enzymes and a substrate before the reaction takes place. So that is why here you have an enzymes without a substrate. If you want to join as a member, just go to my YouTube channel, to any of my video, check, you will see joint. So you click on that and you process it, then you can join as a member. So what we are saying here is we have a substrate plus enzymes, and then the reaction will take place. And enzymes without a substrate, then no reaction. So how do we observe? How do we assay that there is an enzymes? How do we assay that there is an enzymes? We assay that there is an enzymes when we observe a change. A change can be a physical change. It can be in terms of maybe a sound. It can be in terms of a color change. So you have to understand that. Yes, pets, you have. So you need to understand the concept of enzymology. Enzymes, there is a substrate, then there will be reaction, and no substrate, no reaction. And sometimes, apart from the enzymes, we need cofactors. As we are continuing with the lecture, we'll see how the things will work. So now, how do we get enzyme substrate complex? So the substance in bracket reactant and enzymes act on is the substrate. So what an enzymes act on is called a substrate. But in chemistry, we call it a reactant. But in enzymology, the substance that enzymes act on is called a substrate. Assuming now if you have A and B, and A and B converted to produce C and D, if it is in chemistry, the A and B are called reactant, and what you generate at the end of the reaction is called product. But in enzymology or in biochemistry, the reactant is called a substrate. So when you have a substance that enzymes act on, so that substance is called a substrate. And this is how this is how enzymes usually act. So we have an enzymes here and we have the substrate. So the substrate will now join the active site. So it will now enter the clef. It will enter the active site of the enzymes. So after it enter into the active site, now check very well, you can see how you are going to join. Check very well. It's just now that Kehin they join. So just check very well, you will see how to join. Check where you have subscription. Close to it, you will see join. So you see you have a substrate and you have an enzymes. So the substrate usually enter the active site. So the substrate must enter the active site before reaction takes place. Before reaction takes place. So what is an active site? An active site is a restricted region of an enzyme's molecules, or is a restricted region of an enzyme's molecule which bind to the substrate. So it's a region in enzymes where a substrate binds to. So this is the enzymes, and this is the active site, and this is where the substrate binds to. So we have what you call an induced peak. Sometime the active site of the enzymes, do not actually fit with the substrate. So what we are saying here is, if you have an enzyme's active site, so generally the active site of the enzymes doesn't accommodate the substrate. So we have what we call an induced peaks. So in the case of induced fits, a substrate, so it's induced by the substrate. So look at it here. Look at it initially. Look at the active site of the enzymes before the substrate binds. So now look at it as substrate binding. And now it's now changed the shape of the active site. So that is what you call induced fit. That is induced fit. And we have the slide had the cipher. Okay, so let me share again. So I think now you can see the slides. So an induced fit is actually when the active site of the enzymes is changed due to the binding of the substrate. So you have a substrate, and then the substrate, look at it. I hope you have seen how the substrate relates. So you see it is now binding to the active site. So look at how the binding, how the active site of the enzymes change. I hope you see that. Do you all see that? How the substrate change the shape of the active sites. So that is what you call an induced fit. An induced fit model is a model where the substrate bind to the active site and change its shape. And that is what you call induced fit model. So as we are doing the class, as we continue the class, we will see how other models play around, like lock and key model. In the case of lock and key model and enzymes, it's like the theory says that the enzymes is like a padlock. So that is lock and key model. We'll also talk about that. So in the induced pitch model, a change in the configuration of an enzyme's active site, usually hydrogen ion and ionic bonds are involved. So usually in the induced fit model, the configuration of the active site is usually changed, and that change involve the use of hydrogen bond and ionic bonds. Okay, Belikis is asking question. She needs more explanation and clarification on the active site. So you said an active site is like a pocket in the active site. It's like a clip in the enzymes where the substrates pit in, and then the active site will now convert the substrates into product. So it's a region where catalysis takes place. Like for example, in the active site is divided into two. We have what we call catalytic sites. We have catalytic sites and binding sites. A binding site is a site where the substrate binds, and they are all in the active site. And the catalytic site is a region in the active site that catalyzes the substrate after binding. So it means that in the active site, we have also other two sites. We have catalytic sites and we have a binding site. So a binding site is a region where the substrate binds, while catalytic site is a region in the active site where the substrates are converted into product. So that is what we call active sites. So now we are talking about the induced fit model. So in the case of induced fit model, is a model that trying to tell us that when a substrate, sometime the active site and the substrate do not complement, are not complementary to each other, So what happened? In that case, usually the active site, okay, best pair, you are welcome. You are welcome. You are watching us from India, so you are highly welcome and we really appreciate it. Welcome. So in the active side of the enzymes, usually the enzymes do not form complementary with the substrate. So therefore, the substrates will bind to the active site and then change the shape and the configuration of the active site of the enzymes. So once the active site of the enzymes is changed, then the substrate will be perfectly fit into the binding site of the enzymes, and then the catalytic site will now catalyze it to generate the products. And that is what you call enzymes active site and also induced feeds model. So the enzymes do not form complementary, it's not complementing or it's not complementary to the substrates. So the induced feeds model, that's the change in the configuration of the active site of the enzyme is induced by the substrate. So let's look at it. This is an enzymes and the active site. And of course, this is the substrate. So the substrate bind to the active site, and then the now substrate induce the change in configuration of the active sites. So that is what you call induced fit model. That is what you call induced fit model. So there are three factors that affect the activity of the enzymes. What is activity of the enzymes? When we said enzymes activity, it means how much of the enzymes are converted into product. How much of the enzymes or how much of the substrates are converted into product by an enzymes. That is enzymes activity. Some enzymes are very fast while some enzymes are very slow. So when we said enzymes activity, it means is the amount of the substrates that are converted to product by an enzymes. So there are factors that affect the activity of the enzymes. Some factors increase the activity of the enzymes at some conditions or at some certain situation, while some enzymes decrease some factors decrease the activity of that enzymes. And we are now going to look at them one after the other. So number one, we have environmental conditions, we have cofactors and coenzymes, and then lastly, we have enzymes inhibitors. So let's start with the environmental conditions. What are the environmental conditions that affect the activity of the enzymes? So number one, extreme temperature. So usually most of the enzymes work based at body temperature. What is the body temperature of human body? I am asking. What is the human body temperature? I am asking. Okay, you don't know the human body temperature. What is the human body temperature? Okay, so averagely or normal body temperature is around 37 degrees centigrade. So most of the enzymes work at that temperature. And if it is below that 37 degrees centigrade, it means that the enzymes are inactive. If it is below 37 degrees centigrade, the enzymes will become inactive. And if it is above that, then the enzymes will become very active. So if it is above 37 degrees centigrade, then the enzymes will become denatured. So the enzymes will denature. When we said enzymes denaturation, it means when enzymes lose its complete or when it loses its secondary, tertiary, and quaternary structure. So whenever you have an enzyme loses its primary, secondary, tertiary and quaternary structure, it means that that enzyme loses its activity. It means that it will not work again. And that is when I give you this example. If you have a snake venom and you milk venom of snake, and after you allow it to expose to air, because of the environment, there will be an increase in temperature, increase in pH, increase in ionic concentration. So the activity of the venom will be lost. Why is it that it is going to lose? Is because the 3D structure, that is tertiary structure and quaternary structure, are lost. So there will be no activity of that enzymes. And if there is no activity of that enzymes, what will happen? It means that in the case of snake venom, the venom will now remain non-toxic because it's the enzymes that make the snake venom to be toxic. So now in the venom, they are now exposed to the air. So because of that environmental conditions, increase in temperature, ion, and so many other factors, it will affect the 3D structure. So therefore, the activity of the enzymes will be stopped. So that is why he said at extreme temperature, it is very dangerous. It is very dangerous because it affects the activity of the enzymes. So ladies and gentlemen, we should understand that temperature plays a crucial role. It plays a crucial role in maintaining the activity of an enzyme. So that is why it is dangerous if the temperature is above the normal body temperature. So remember, below that 7 degrees centigrade, the enzymes is inactive, and above that, the enzymes will become denatured completely. So it will lose its 3D structure and no function. So the temperature must be within 37 degrees centigrade. I hope it's clear. So then the next thing is high temperature may denature, that is, it will now unfold the enzymes. So it will unfold the enzyme. So the next thing is pH. Most like 6 to 8 pH, that is near neutral. So you should understand that in the case of pH, I think, ladies and gentlemen, we have talked about pH. The lower the pH, the higher the acidity, and the higher the pH, the higher the basicity in terms of alkaline or in terms of alkali. So most of the enzymes work best at a at a pH between 6 to 8. But there are some extension. There are some, it's not in all cases. We have a situation where the pH is below that and there are enzymes and they are active. Like in the mouth, we have an enzyme called salivary alpha amylase. And salivary alpha amylase is an enzyme that found in the saliva. And the pH of the mouth or of the saliva is around 7, that is neutral. So if you look at the pH here, it falls within the range of the activity of salivary alpha amylase. So, but now the question is, what about in the stomach? We have enzymes, pepsin, and stomach is highly acidic. In fact, the pH of stomach is within a range of 1.0 to 2.5. So it is highly acidic. So there is an exception. But most of the enzymes in the human body, they work best between 6 to 8 pH. So you should understand that. Okay, good day, Grace. Thank you for joining as a member. So these are actually some of the things that we should look from the environmental conditions. And there is also ionic concentration, that is salt ions, that is salt concentration. It also affects the activity of the enzymes. So the higher the ionic strength, or the higher the ionic concentration, the lower the activity. So we should understand that the ionic concentration should always be maintained within the required range. So what are the cofactors? So we have cofactors and enzymes. So cofactors are inorganic substances. Cofactors are inorganic substances that are found in the enzymes. So cofactors are inorganic substances like zinc or iron, and they are found in the enzymes. So we call them cofactors. And the coenzymes are vitamins and are sometimes needed for proper enzymes activities or protein in general. So there are some enzymes that require cofactors and coenzymes before they carry out their enzymatic activity. What is enzymatic activity? It means converting substrates to product. And you need these cofactors and coenzymes. But it's not every enzyme that needs that. And let me just give example for the enzymes. So in the case of iron, iron is an example of inorganic substances that are present in the quaternary structure of hemoglobin in order for it to pick off oxygen. So if there is no iron, there will be no functions of the hemoglobin. In fact, there will not be even the hemoglobin. So that is a cofactor. It's an inorganic minerals that is found in the hemoglobin for the hemoglobin to function or for the hemoglobin to work. So then we have two examples of inhibitors. So what are inhibitors? Although the examples of the inhibitors are more than this. Yeah, there are more than this. And we are going to look at them. This is just to give you a general introduction of the enzymes. The purpose of this lecture today is for you to have a basic understanding of the concept of enzymes. So we have two examples of enzymes inhibitors. Here, we have competitive inhibitors. So competitive inhibitors are the chemicals that resemble an enzyme's normal substrates. And therefore they compete with the substrates for the active sites. So when you say competitive inhibitors, it means that the substrates and the enzymes, and the inhibitor, they look alike. They look the same. So what happened in that case? The substrates will be competing the active site with the inhibitors. So therefore, if the inhibitors bind to the active site, it means that there is no point or there is no active site for the substrate to bind. So it means that there will be no catalysis. So most of the drugs that we generated, most of the drugs that are discovered and generated in the pharmaceutical industry, they are competitive inhibitors. Because there are some enzymes that are important for the survival of a particular microorganisms. So if you are able to identify such enzymes, you can design an inhibitor that can bind to the active site of the enzymes and block it. And that is what we call enzymes inhibitors. Sorry, that is what we call competitive inhibitors. Like for example, some enzymes that are important for the pathogenesis of a particular disease. So if you block the activity of that enzyme, it means that you are blocking. Like for example, let me just give this example. Remember we have what we call angiotensin converting enzyme. is an enzyme that produce or that convert angiotensin 1 to angiotensin 2, which is a vasoconstrictor. So it causes or it leads to a disease condition called hypertension. Yeah, because it's a vasoconstrictor. So it constricts the arteries. So if you are able to identify a compound that can block the activity of, that is why in most cases, especially in hypertension, we'll realize that it's due to activity of that angiotensin converting enzymes because it generates or it reduces a lot of vasoconstrictor, and that is why there is that hypertension. So if you can get any drugs that can slow the activity of that particular enzyme, or if you can design any chemical molecules that can inhibit the activity of that enzymes, what will happen? It means that it will lower the activity and there will be less generation of vasoconstrictor, and of course it will be as an agent that will slow the pathogenesis of hypertension. And in some microorganisms, you have some microorganisms that some of the enzymes is important for their survival. So if you block the activity of such enzymes by designing a competitive inhibitor, then you can kill the parasite or you can kill the organisms. So this is what we do in drug discovery. So you know that you are going to do group assignment, and this one will be one of this concept will be one of the assignment for one of the group. Okay, is there anybody that want to work in this? Who want to work on the drug design discovery using enzymes? Is there anybody that is interested? I'm waiting for your response. Who want to work? Okay. Oluwat Oluwatosin, do you want to work? Do you want to do your group assignment on the application of enzymes in drug discovery? Okay, so thank you, no problem. I will give you that. So you and your group members will work on this. But Oluwatosin, you didn't join the membership to this channel. So that is that. So that is for the competitive inhibitors. So then the next thing, so look at it, this is an enzymes and this is the competitive inhibitor. So look at it, the inhibitor is perfectly fit into the active site of the enzymes. Is perfectly fit into the active site of the enzymes. And this is a typical example of competitive inhibitors. And of course, look at the substrate. So there is no pocket. There is no pocket for the substrate to bind. And then the other one is non-competitive inhibitors. So these kind of inhibitors that do not enter the active site but bind to another part of the enzymes, causing the enzymes to change its shape, which in turn alters the active site. So you see, in this case, in the case of non-competitive inhibitors, the substrates and the inhibitor is not binding to the active site of the enzymes. Rather, it is binding to another site on the enzymes rather than the active site. And once it binds, then it will now change the conformation of the active site. If your own is not working, you can try any bank. It might not be only your own. You can use any other person. So that is for the non-competitive inhibition. Or you can even try to send tanks here. There is one tanks there in dollar. You can try that one and see. So usually that's what happens. So the inhibitors, they do not actually bind to the active site. Rather, they will just actually try to check the binding site. Sorry, try to actually bind to another site. And then once it binds to the other side rather than the active site, they will change the conformation of the active site and the substrate cannot bind to it. So you see, this is the enzymes, and this is a competitive inhibitor. So you see, the non-competitive inhibitor bind, and then the active site is altered. Remember, this is the active site before. Look at it, but because after the substrates, also after the non-competitive inhibitor bind to the other sites on the enzymes, then the substrate couldn't bind because the active site is altered. So that is what happened in the case of non-competitive inhibitors. So this is actually what we what you need to know for the basic concept of these non-competitive inhibitors. And okay, Saeed